Welcome to the first of our 3 speakers this evening. Please welcome Lisa Toudic, who is presenting her work on the assessment of the epi-chaperone in acute myeloid leukemia at this year's Westchester Science and Engineering Fair on March 16th at Sleepy Hollow High School. Lisa was awarded the Innovations in Biological Sciences Research Award. For all of her hard work and dedication. So we're very proud of her, and please take it away and enjoy.
Hi guys, welcome to my presentation on the assessment of the epi-chaperone in acute myeloid leukemia. And I promise that it will get much more simple. So to start off, what is acute myeloid leukemia? So for short, it's called AML, and it's a blood cancer. It's a type of leukemia which is a blood cancer, and it is formed through this process. So as we all know, a stem cell is an undifferentiated cell that further differentiates into any cell. So basically, it can become whatever it wants in the body. And through the process of differentiation, in this case, it becomes a blood cell. However, during this process, it acquires a mutation. And this makes it so that the stem cell cannot further differentiate, and it accumulates into the cancer. It is categorized as the rapid growth, and it is also interfering with normal blood cell growth, and is also mostly seen in older patients. So these are the different treatment options for leukemia. For example, chemotherapy, targeted drug therapy, or transplant. And as you can see in this diagram, these are the diagrams that doctors use when they decide what to give the patients. And the patients that I looked at went through this whole process and did not respond to any of them. So basically, we really wanted to know why these patients did not respond to any treatment. So Guzman and Cayosis 2016 also asked this question of why, and they discovered a protein folding complex called the epi-chaperone. And this is basically just a complex inside of a cancer cell. It is made up of 2 types of cells, HSPs, which are heat shock proteins, which come about when cells are under a lot of stress, meaning treatment. And notably, one type of HSP that we found constantly in the epi-chaperone, which HSP90. Then there are also chaperone proteins. These proteins basically fold other proteins to their proper shape so that they can function. However, in the case of the epi-chaperone, they allow cancer-inducing proteins to function through any type of treatment, which is why these patients did not respond to treatment previously. So Guzman and Cayosis hypothesized 2 basic functions. They hypothesized a way called the novel binding assay, and this is a big word for binding the epi-chaperone. This process uses a PU50 compound, which is essentially a dye that enters the cell. So for example here, we can see the PU50 entering the cell in a positive epi-chaperone cell. As you can see, the epi-chaperone is present. Once it infiltrates, it becomes extremely green, and this tells us that the cell has the epi-chaperone. However, in a negative epi-chaperone cell, meaning there's no epi-chaperone, it's a normal cancer cell, when it infiltrates, there's little to no binding of the PU50 compound. They also hypothesized a way, after they found whether or not cells had the epi-chaperone, to target the epi-chaperone, meaning eliminate it from the cell. And this is done through targeting or taking out HSP90, which we saw before, from the epi-chaperone. And it works because the epi-chaperone is a complex that is so intertwined together that when one piece of it is eliminated, it completely dissolves. So as you can see, it infiltrates the cell, binds to that little HSP90 cell, completely eliminates it, dissolves, and gives us a dead cell. So my questions and goals for this study was to determine whether AML cells had the epi-chaperone through the first assay that we found, and then successfully eliminate the cells that are reliant to the epi-chaperone through targeting it with PUH71. My hypothesis was that the cells that we found epi-chaperone positive through the binding assay would be most susceptible or sensitive to the treatment that we would use after. So the methods, or the way that we conducted this experiment, was done in 2 specific layouts. First, the binding assay that, as we know, finds whether or not cells have the epi-chaperone, and the second, which is the viability assay, which eliminates the epi-chaperone. So the novel binding assay methods included patient samples, notably 11 primary patient samples, which all were relapse refractory AML, meaning they were just not responding to any treatment. Then we also used cell line controls, and these were used to compare our data to, because we knew that MB411, we hypothesized that it was epichaperone positive, meaning having the epichaperone, and HL60 was hypothesized epichaperone negative. Then this is the plating layout that I used. As you can see, I left the first row untreated or unstained, and this was done to compare the samples to whether or not there was a substantial error. Then the second row was stained with PU-FITC, which, as we know, finds the epichaperone in the cell. And the third one is a rather unusual one. It actually uses the FITC9 compound, which binds to anything not epichaperone positive. So it's basically our background probe. So we used a machine called the flow cytometer through this experiment, and we analyzed things through the FITC channel, which allowed us to see the fluorescence of each probe. So as you can see, the first peak represents the FITC9 probe, and the second peak shows the PU-FITC, which finds the epichaperone. We also stained the samples with antibodies, which basically shows us different functions in the flow cytometer. Then we use the gating of cells. The gating of cells basically sections specific cells that we want to analyze. It has several processes. So we started off by sectioning live cells from debris. Then we sectioned them off, for example, from live and dead cells. And then we further sectioned them from cell type. And this allowed us to analyze specific cells. Here is an example of the broken down version of the FITC channels that we would see. And this allows us to analyze the samples. So as you can see, the first one finds the epichaperone, the second one is the background, and here is the overlap that we saw before. And this overlap gives us a key function right here. This is the binding, which is the overlap between the two peaks. This binding tells us whether or not samples has the epichaperone. It was analyzed through calculations using lymphocytes, which were used as a control because they're regular white blood cells, and blasts, which are cancerous cells. The calculations included finding the mean fluorescence intensity, meaning the mean fluorescence through each probe. And then we analyzed through lymphocytes and blasts. We subtracted the PU-FITC with the background FITC9, and then divided them by blasts and lymphocytes. That might be a lot of information for just finding a specific number, and this number tells us whether or not samples have the epichaperone. It was concluded by Guzman and Kiosis that if the unit that we found was above 2, this had samples positive epichaperone, meaning the samples had the epichaperone. If it was lower than 2, this meant that the samples were epichaperone negative, or no epichaperone was in them. So the viability assay, which as we know from before, targets the epichaperone or takes it out of cells, used 5 of the samples that we tested in the previous assay. This is the reason because we had a lack of resources from the binding assay. And in this case, we used lymphocytes as our control because they are not cancerous cells, meaning they would not have the epichaperone, hopefully. We hope that they don't. We use them because they would not respond to treatment. So the layout is pretty similar to the other one. It uses a background row with unstained, and then the second one was treated with our PUH71 compound of 0.5 micromolars. So we also stained them through antibodies and passed them through the flow cytometer, which gave us different functions of the cell. And then we also used the process of gating cells with lesser steps. So for example, we only gated them from live and debris cells and then live and dead cells. So in these calculations, we analyzed for a % decrease from cells that were not treated with the compound to cells that were treated. So for our results, I also broke it down into 2 sections. So our results for whether or not samples had the epichaperone, and then our results for whether or not they responded to the treatment. So the binding assay results found that, as you can see, the blue dots represent all positive epichaperone cells, and then all red dots show that the samples that we found were negative. So 6 of the 11 samples that we tested were epichaperone positive, as you can see, because they have a count higher than 2. And then 5 of them were concluded epichaperone negative, which, as you can see, are the red ones and have a count lower than 2. As you can see here are cell line controls, And as you know before, HL60 was hypothesized negative, and we found that our hypothesis was correct because we found that it was negative in the end. And then MB411, which was hypothesized positive epichaperone, had a count higher than 2, which means that it was positive epichaperone. So the viability assay results. Here are the samples laid out that we analyzed in the viability. So as you can see, here are the 5 of them, and only 4 of them were epichaperone positive, and 1 of them was epichaperone negative. Here is a lot of information, and I will break it down. As you can see, for example, AML 147 previously was concluded to be epichaperone positive. And as you can see, it started off with a viability of 34.10 before it was treated, and after it was treated, it decreased significantly to 8.10 viability, meaning that the treatment did work. However, for AML 140, as you can see right here, it's negative. So when we saw the results, we saw that it had a very low decrease from 76.5 to only 71.8, meaning that it did not respond, meaning that our hypothesis was correct. So the conclusion of the 2 assays ended up being that the levels of the Pu-Fitzy fluorescence does tell us whether or not samples have the epichaperone. And this successfully identifies whether or not samples have the epichaperone so that we could further eliminate it through the second assay. And then this is an example of precision medicine and not mutation-based medicine, which is very novel, and we are very excited about it. For the viability assay conclusion, we found that the samples that were positive epichaperone did respond to treatment, meaning that we were correct, and if samples had a high abundance of Pu-Fitzy, they responded the best to treatment, meaning if they had a count extremely higher than 2. So these results successfully led to a compassionate trial. And a compassionate trial is basically taking one patient who has no more chances at survival and giving them a chance. So this compassionate trial, we only analyzed one patient and we gave them this treatment because it was their last case scenario at survival. So the compassionate trial was allowed because we saw that the drug PUH71 was previously used, so we knew that it was not too toxic and it would allow them to respond successfully and it wouldn't hurt patients. So we found that the patient that we analyzed did have a high abundance of epichaperone in their cells, which allowed us to analyze them through the PUH71. And this is an example of testing the treatment through in vivo. And in vivo means in life. So this was the first time that we tested things in life. So as you can see, here are results overall, and even though it's very confusing, I will break it down. So as you can see, before treatment, the patient had a lot of these purple, kind of larger cells, and this is the cancer. And after treatment, we found that it had normal blood, little to no large blasts like these, which means that the patient did respond to treatment, meaning that our drug could be used for other patients once it's tested further through mice and through all of that, but it will work in the end. So for future research, this summer, I will want to analyze the toxicity of this treatment, meaning whether or not it is safe for patients to use long-term. I would also like to analyze my data further because, as you can see before, I would like to see if patients with a high abundance of 2 would respond better to treatment rather than the ones that had a lower count of 2. And I would also like to find ways to reduce the amount of PUH71 we're giving to treatments to really reduce the amount of toxicity there are in their body. So here are my references, and this was my project on the assessment of the epichaperone in acute mild leukemia. Thank you for watching. I hope it wasn't too confusing. Do you have any questions?